wanted to thank the Pasteur Institute to invite us to this workshop. And it was very interesting to see there's a lot of mass spec and GCMS work, and of course we're kind of the opposite of that, and therefore it's kind of interesting to present that technology, which is kind of perhaps a bit anterior to GCMS and so on, but, and it's also interesting to see the future of that technology, and maybe we can discuss that at the roundtable afterwards. I'm just going to introduce Biospace Lab a little bit. We are a company that actually I've just realized that we're celebrating an anniversary with the Pasteur Institute. Pasteur Institute is celebrating its 130th anniversary. We're celebrating our 30th anniversary. The company was started by the Nobel Prize for Physics George Charpak, and we have an expertise in autoradiography for the last 30 years, and also in scintigraphy, and more recently in optical imaging. We have systems installed worldwide, about 300 system base installed worldwide, and we have two branches of work of systems. We have the in vivo imaging system, which is optical, and the ex vivo system, which is the autoradiography systems, which separates into two branches: the high-speed, high-throughput digital imager, BetaIMAGER T-Racer system, and the high-resolution BetaIMAGER D5. So I'm going to start with the BetaIMAGER T-Racer. Both systems are based on the same technology of photon multiplier coupled to CCDs that allow real-time imaging by photo counting, creating list mode file data, and the temporal information can be used to improve on quantification. I'll get into this a little bit more in detail later. The quality of the photon multiplication allows us to have extremely low detection thresholds and an accuracy, a quantitative accuracy over 5 logs of magnitude, so very linear in terms of quantification compared to other autoradiography technologies. And we can do a field of view with this system of 20 by 25 centimeters. Okay, so the technology is based on the multi-wire avalanche chamber of George Charpak, for which he got his Nobel Prize. And then with a scintillation based on gas scintillation with a mixture of a solvent, tritilamine, which will scintillate into photons, which will be collected by a camera, photon multiplied in a photon multiplier tube before collecting in real time by the system. The importance of this amplification through the avalanche chamber and through the photon multiplier gives us a time resolution of hours instead of weeks for low energy isotopes. So we can image tritium in cryosections in 12 hours instead of 12 weeks for film exposition. And this is really has been our USP, if you'd like, our unique selling point is the speed of acquisition for which pharmaceutical companies all over the world have purchased for systems in order to have fast resolution of their experiments. Okay, the BetaIMAGER also has zoom capabilities to increase on spatial resolution, and this is a zoom, a mechanical zoom on a full field of view to increase on the quality of the images that you would get from a full field. The BetaIMAGER T-Racer is slightly restricted in spatial resolution compared to film. It has a resolution of 50 micrometers for low energy isotopes, and about 200 micrometers for high energy isotopes. So the image is not of the quality of film, and we know that. And this is why we have developed the BetaIMAGER D5, which is high resolution. We used to call it the micro imager. This is how probably the Pasteur Institute knows it, because they have one of each systems. So the BetaIMAGER D5 is high resolution, and it can go down to 10 micrometer spatial resolution, which is really to the standards of film. But it still managed to do the real-time imaging with extremely low detection threshold and a very good dynamic range of 10 orders of magnitude. And also, we can use the real-time technology to separate multiple emitters in the same sample. Multiplexing imaging is a strong advantage of those systems. Okay, so the technology of the Define is much more simple than the BetaIMAGER. We completely remove the avalanche chamber and we're just focusing on the photon multiplier tube connected to the CCD, and the scintillation is not gas anymore, but it's a solid scintillation foil that is applied to the samples, often cryosections laid on histological slices. Okay. So the BetaIMAGER Define gives us resolution that is very much equivalent to film, but with a better order of magnitude. So you can see the levels of the gray levels are much better resolved, so you have a much more quantifiable information compared to film, which saturates very quickly. And that's one of the other things that is good with digital imager systems, is that you cannot oversaturate, you cannot saturate the exposition, you cannot overexpose and get saturated values, because actually the photo counting is done every 30 milliseconds at the speed of the frame renewal of the camera. So another advantage when I was talking about multiplexing or multi-labeling, so we can use the temporal information of the decay of various isotopes to actually separate the signal. And here we're looking at F18 FDG compared to technetium 99, and we can separate the information from one image into two images thanks to the time profile decay. So you can see that if you're looking at the last images around 10 minutes, most of the signal from F18 has completely decayed, so the images that you get around this period will be mostly linked to technetium 99. And this is how we do frame by frame, pixel by pixel by pixel. We separate the information from the two isotopes, and we can reconstruct the image afterwards, creating multi-label imaging. And this is an example of biological information collected from multiple imaging, with F18 showing glycolytic effects, technetium 99 showing perfusion, and indium 99 showing necrofibrotic areas. And you can see that wherever you have fibrotic areas, glucose consumption is reduced and perfusion is reduced as well. Okay, so coming back to what's interesting about the BetaIMAGER systems and the advantage of autoradiography perhaps in biochemistry or a study of protein, lipids, and glucose, is that there is no label steric hindrance with isotope labeling, and therefore, compared to labeling of compounds with perhaps fluorescence molecules, for example, the affinity of the target will never be affected by the steric hindrance of the label using autoradiography. They also perhaps arguably now, the unmatched sensitivity of radioactivity assays, and the fact that you can go down in very low molecular amounts, and this is why it's been used, for example, in Western blots and thin layer chromatography to monitor low levels of tracers in tissue samples or in cell lysates. Another, again, arguable advantage of autoradiography is that some of the labeling can be fairly simple. So metabolic labeling of proteins or lipids in cells is pretty much just a case of adding a compound based tritium, which is labeled with a radio label, and let the cell take it up, and then we can have, with various biochemical methods, identify the metabolites of those compounds. I'll show you some example of that. The digital autoradiography, which is linked to the BetaIMAGER system, has extra benefits. The speed of acquisition, which compared to normal autoradiography is minutes over weeks, And there is no blind processing, so no over or under exposure, so you always know what you're getting. Here's an example of what you see for an acquisition of over several minutes. You can see that the signal is progressing in time, and you can see that in real time in the window of your images. So you can actually see whether you have incubated enough or imaged enough or not imaged enough or carry on imaging to get the best image. And the more you will image, the better the quality of your image. But you can also come back to the original data and do a temporal window from the acquisition to look at what the image would look like for a smaller amount of time. So that's a way of improving on your workflow and making your acquisition less long, considering that you can look at the data of a file that would be acquired over several hours, and then decide what would it look like after 1 minute, 5 minutes post-acquisition. So that's done in the analysis software afterwards. Okay, we can do multi-labeling as I showed you, and we can also look at decay of compounds such as F18, FDG, and so on. You can look at the decay of the signal, which can be used to help for quantification as well. Okay. As you know, the drawback of autoradiography is the management of radioactive material, especially for low energy isotopes. The more and more restrictive regulation over storage, usage, and disposal of radioactive compounds. And for these reasons, I would say a loss of expertise in the scientific world, because people are scared to use radioactivity more and more, or they will use alternatives. And therefore, there's a loss of expertise of people that know how to do autoradiography well. Okay, so other advantages of the autoradiography methods is that, and the BetaIMAGER system, is that you can use various types of samples. You can look at tissue sections. You can look at quantitative WBA, whole body biodistribution analysis, such as these ones. You can look at thin layer chromatography for lipid, to study lipid composition. You can do 2D TLC analysis. You can do Western blotting to look at protein and glycoprotein analysis. You can do immunoprecipitation, pull down assays to look at protein and protein interactions or protein-lipid interactions. You can also do 2D electrophoresis or native gel analysis. And you can also do in situ hybridization to look at protein expression or biodistribution of protein expression. You can also look at microarray to do a transcriptome analysis over a very small amount of space. I'll show you some data about that later. So there are various compounds that can be used with the BetaIMAGER, not only the direct beta emitting, but also the indirectly gamma emitters, and some alpha emitters can be imaged with the BetaIMAGER as well. And of course, you can image the positron emitters that are for PET validation, for example. Okay, so this is an example of the quality of image you will get from a whole body quantitative analysis, which is really, today, the autoradiography for QWUVA is a gold standard for DMPK studies in the pharmaceutical world. And the resolution of the image and the quality of the image here with the quantitative information as well with the coded lookup tables. Very quickly, because I don't want to talk too much about neurosciences, but the main application of our system in the pharmaceutical industry are linked to ligand binding assay, receptor occupancy, and in situ hybridization. And this is a paper where you can look at the biodistribution of beta amyloid plates with high resolution in brain sections, and validate PET information with high resolution using the BetaIMAGER define. You can also, here's an example of multiple labeling where they're using two different peptides linked to beta amyloid plates, to label beta amyloid plates linked to F18 or tritium. And this is the one image, and this is the extraction of the post isotopes from the one image collected. and the validation of the information by immunohistochemistry as well. This is an experiment, a recent experiment done by Imanova at Imperial College London, at Hammersmith Hospital, where trying to move on from neurosciences now, where you can look at specific peptide for the translocator protein in control versus TPSO knockout routes. And what I find very interesting in this paper really is the use of embryo microinjection of zinc finger nucleases to actually perform the knockouts on those mice. And then they can look at the biodistribution of the specific PK peptide in the adrenal gland in wild type versus knockdown mice organs. Another application for labeling of organs. So here you've got DAMSA to label the kidney, and also phantopeptide labeled with lithium. So this is moving on to closer to what people do here. You can be looking at biometabolism of proteins, lipid, and glucids. You can look at the expression of protein using by transcriptome analysis using microarrays. And these are microarrays that was developed in the Institute Paoli Calmette in Marseille, where they can look at 6,000 DNA sets with a 225 micrometer pitch. So each spot is separated by 225 micron, and they can look at different labels, hybridization of the gene map. And you can also do that, and this was done at the hospitals PTS-Salpêtrière. You can actually do differential screening microarrays, where you're actually taking two probes and you're putting together and letting them hybridize together. And then with the separation capabilities of the BetaIMAGER define, you can separate the results and quantify them by looking at the differential expression of both, or hybridization of both peptides. So moving from, we can also do high in situ hybridization, and here's a S35 labeled probe specific to actin mRNA, and looking at the biodistribution of actin in mouse embryo. And you can see here a resolution of 15 micron between the different fingers of the actual embryo. So define really high resolution and high quality images. And here, getting again closer to what you're doing here, this is an example of how you can use the BetaIMAGER T-Racer to look at Western blots from pull-downs assays. This was a study that was done recently, an old study done at the University of Genova, where they looked at fucosylation of Notch1 in pull-down assays. And they looked at human fibroblasts labeled with treated fucose, and the human fibroblasts were taken from patients with leukocyte adhesion deficiency type 2 or control patients. And you can see that in the intracellular domain of Notch1 pull downs, there is no difference in fucosylation of Notch1, but in the extracellular domain, which is slightly heavier, which is pulled down, you can see a difference in fucosylation in the lab 2 patients. Now, if you carry on a further removal of the N-linked oligosaccharide using the PGNSF enzyme, you can see that the actual oliglycans are not affected, thus suggesting that the decrease in fucosylation in lab 2 patients is due to N-linked fucosylation. This is a very old paper. I'm sure there's lots of arguments about this since, and there's been quite a lot. But there you go. This was done with the BetaIMAGER. Right. Moving on to membrane protein-lipid interaction again with Western blots and pull-down assays, and you can see here that. We're looking at, it's a Finnish team from Stockholm University who developed a motif probability analysis tool, MoPro, to actually identify transmembrane helix motifs specific for sphingolipid binding. And what they did is they found about 28 motifs expressed in 900 proteins, and they selected 4 of those for, in this particular paper, for autoradiography analysis and pull-down assays. Each of these motifs being expressed at different membrane compartments, so the TGN, the ER, the PM, the plasma membrane. And here they're using SHGR1 as a control, which doesn't have any sphingolipid binding motifs. And you can see that they're looking at the radioactivity in control versus specific binders, peptide motifs, and you can see a 3 to 12-fold increase in sphingolipid binding after metabolic labeling with tritiated photosphingosine. A more recent paper, this one, by the Italian team in Milan, who's doing a lot of sphingolipid work using the BetaIMAGER, and here is looking at sucrose-loaded fibroblasts as a model of lysosomal storage disease, and they're looking at the catabolism of H3 sphingosine GM3. So they're loading the cells, and they can see that in sucrose-loaded fibroblasts, the catabolism of GM3 into ceramide, glycosyl ceramide, and lactosyl ceramide is decreased, whilst GM3 is the GM3 load is the same. So just looking at the different catabolism of lipids using the autoradiography and thin layer chromatography to separate the lipids. Okay, so this is the same team looking at a high performance thin layer chromatography now, again to monitor synthesis of sphingosine 1-phosphate S1P, the effect of mesenchymal stem cells from patients, lipoaspirate. So what they do is they take some fat from patients, and then they either give it or co-incubate with microbial cell cultures, or they purify the ADSC cells using lipogen methodology, and then they look at the effect of those ADSC cells on the production of LPS, on a model of LPS stimulation. So here they can show that the secretome inhibits LPS-induced production of 3-HS1B in microglial cell cultures. So here the control, and then the cells treated with the stem cells from patients, and you can see a reduction of the LPS of the production of S1P. I mean, quickly, it's my last 2 slides. So very quickly here is just an example of measuring how to measure glucose metabolism using F18-2DG. You probably don't have any interest in that. So I'll move on. Interest, again, probably no interest, very beautiful images of sucrose uptake in Arabidopsis, so looking at sucrose uptake. And here I wanted to finish with this very beautiful paper published here at the Pasteur Institute using the BetaIMAGERs of the in the laboratory of the late Anna Cordova, and it was a very interesting paper where they look at peptidoglycan uptake in epithelial cells, and what they did is they had a mutant deficient in the guide pathogenicity island gene responsible for type 4 secretion, and by knocking down this gene, they could see a decrease in uptake of PEG, of peptidoglycan labeled with tritiated DAD. And they could correlate the information that they quantified from histological slides, histological slides on which the epithelial cells have been grown by looking at histo, so taken with the microimager and correlating it with the overlaying it with the information from the histological staining. That's it. And from that, they could conclude that from other experiments in the paper, we can look at the involvement of Nod1 to regulate NF-kappa, the pro-inflammatory response. And that's it. So just to show you the number of systems installed worldwide, and if you have any questions, please do not hesitate.